So heredity. So for the genetics, we start with the father genetic law. So first law, second law, and third law. Now after this, we talk about nowadays genetics theory. Okay, so now let's start with general concept. Okay, so we already talk about homozygous, right? And heterozygous, right? So, and alleles and the genes. So everyone knows genes, right? So we will talk about genotype, phenotypes, dominant and recessive. Okay. So now we're going to start a video. So give you an idea what's this concept for, because those concepts are really fundamental for genetics. We have to remember this. So now let's start with this one. Give me 2 seconds. Let me pull out the video for you.
Hi and welcome back to freesciencelessons.co.uk. By the end of this video, you should be able to describe what's meant by dominant and recessive alleles. You should then be able to describe what's meant by homozygous, heterozygous, genotype and phenotype. Now this video does contain a lot of definitions and it's really important that you learn them. I would suggest that you watch this video several times until you're happy with the ideas. In previous videos, we've seen that DNA is found in chromosomes, and I'm showing you here a Hi and welcome back to freesciencelessons.co.uk. By the end of this video, you should be able to describe what's meant by dominant and recessive alleles. You should then be able to describe what's meant by homozygous, heterozygous, genotype and phenotype. Now this video does contain a lot of definitions and it's really important that you learn them. I would suggest that you watch this video several times until you're happy with the ideas. In previous videos, we've seen that DNA is found in chromosomes, and I'm showing you here a human chromosome. Remember that normal human cells contain 23 pairs of chromosomes. So that means that normal human cells contain 2 copies of every chromosome. And I'm showing you that here. Now a key fact that you need to learn is that one chromosome in the pair comes from your father, and the other chromosome comes from your mother. We also saw that genes are sections of DNA on a chromosome. And again, because chromosomes come in pairs, we've got 2 copies of every gene. I'm showing you here the gene which controls earwax. We're looking at the earwax gene because earwax is controlled by a single gene. Most characteristics are controlled by many genes acting together. Now, genes often come in different versions, and scientists call these alleles. So alleles are versions of a gene, and that's a key definition that you need to learn. The gene for earwax has 2 alleles. One allele leads to wet earwax, and we're going to give this allele the symbol capital E. The other allele leads to dry earwax, and we're going to give this allele the symbol lowercase e. As we said before, we've got 2 copies of every gene. So imagine that a person has 2 copies of the allele for dry earwax. I'm showing that here. The genotype of a person tells us the alleles present. And again, that's a definition you need to learn. So this person's genotype is lowercase e. Because this person has 2 copies of the same allele, scientists say they are homozygous. Okay, now the phenotype of a person tells us the characteristics caused by the person's alleles. So because this person has 2 alleles for dry earwax, their phenotype is dry earwax. Okay, this person has 2 alleles for wet earwax, so their genotype is capital E, capital E. And again, because both alleles are the same, they're homozygous. So because this person has two alleles for wet earwax, their phenotype is wet earwax. Okay, here's a third person. Their genotype is capital E, lowercase e. In other words, they've got one allele for wet earwax and one allele for dry earwax. Now in this case, the person has two different alleles. Scientists call two different alleles heterozygous. So this person is heterozygous for the earwax alleles. So what phenotype will this person have? Well, in the case of earwax, the allele for wet earwax is dominant to the allele for dry earwax. A dominant allele will show in the phenotype even if there's only one copy present. So in this case, because the allele for wet earwax is dominant, this person's phenotype is wet earwax. The allele for dry earwax is recessive. A recessive allele will only show in the phenotype if two copies are present. In other words, if no dominant allele is present. As we saw before, this person has two copies of the allele for dry earwax. So because there's no dominant allele present, their phenotype is dry earwax. Now, as we said at the start, some characteristics are controlled by a single gene. However, most characteristics are the result of many genes acting together, for example, height. In the next video, we're going to look at genetic crosses and see how alleles are inherited. Remember, you'll find plenty of questions.
Okay, so now let's back to this page. So this page are extremely important for you to review all of general concept. Okay, please watch the video several times if you have problem with those concept. So now let's start with first law. The first law of the genetics is the law of the dominant. In further genetic time, they do not have a concept of alleles, so they use this name traits. Okay, so the traits. So because we talk about we're linked to modern genetics, so in this class we'll use alleles. Okay, so allele, let's say you have two parents. So you have two alleles. Okay, so the two alleles, each one can be dominant or they can be recessive. So if you have a dominant one, then they will show dominant. Okay, so if you have a recessive one, if you one of your parents gave you a recessive one, they will show probably recessive or dominant, depending on another one. If another one is dominant, okay, you follow the dominant, you show dominant. If another one is recessive, you show recessive. Okay, good. So now let's give you examples. So order of biology is by examples. Okay, so now let's see here. So you have two parents. So one is very tall, another one is very short. So when you cross these two, then you actually see this one? You will have here is very tall. Okay, so this can best illustrate by the Punnett square. So how you do Punnett square? I will show you. Okay, I will give you another short video you can watch. So to learn genetics, number one, you have to know the concept we just talked about. Number two, you have to do Punnett square. Okay, someone say my brain is very clever, so I don't need to do Punnett square. So I guarantee you, sometimes you will make a mistake. If you do Punnett square, they will make sure that you get 100% chance correct. Okay, do Punnett square. Punnett square is very important and very simple to do. Okay, so now let's look at Punnett square.
Thanks for stopping by. Today we're talking about the basics of Punnett squares. Punnett squares measure the probability or likelihood of a genetic outcome based on a specific genetic cross. It doesn't tell you what will be, only what could be. For the simplest of Punnett squares, you'll cross two genotypes. These parent genotypes will be for the same gene but may or may not contain different alleles. Let's look at an example of a genetic cross for eye color. Green eyes being dominant and blue eyes being recessive. And we'll use big G, little g for the maternal genotype and little g for the paternal. So this is our cross. The mother has green eyes and the father has blue eyes. And we want to see the probability that their offspring will have either green or blue eyes. For this cross, we'll draw a square split into 4 quarters, like a simple 4-panel window. This is the Punnett square. One of the parental genotypes will go on top and the other will go on the side. It doesn't matter which genotype goes where, and the order of the letters really doesn't matter, but we conventionally write the dominant allele first, if there is a dominant allele. So your setup should look like this. Then you simply carry the letters through to their respective quadrants and match them up, like so. All right, each quadrant will represent a 25% chance that that genotype will result in the offspring. So in this case, we have only 2 possible genotypes, big G, and little g, and each genotype has a 50% chance of showing up in the offspring. Similarly, you have 2 possible phenotypes, green eyes and blue eyes, with a 50% chance of each. Let's look at another example. We'll cross 2 genes that are heterozygous for eye color in this example. So big G crossed with big G. These colors are just for illustration purposes. So we again will draw our Punnett square and put one gene on top and the other down the side, and then carry the letters through to their respective quadrants and analyze the results. So this cross results in a 1 to 2 to 1 genotype ratio, with 25% homozygous dominant, or big G, 50% heterozygous, or big G, and 25% homozygous recessive, or little g. The phenotypic ratio would be a 3 to 1 ratio, with 75% green eyes and 25% blue eyes. Okay, hopefully that helps you understand simple Punnett squares a little better.
Okay, so let's do Punnett square for this these trees. So your father, let's say your father is a big T, so they show very tall. Your mother is short, they show small t. They're both homozygous. So when you cross, so your big T combined with small t become big T, small t. So you can see you only have 1 genotypes and also 1 phenotypes too. So the phenotypes is big, tall. The genotypes is big T, small t. Okay, so the phenotypes and the genotypes. Okay, so now let's talk about second law. So what's the second law? The second law is a law of segregation. So what's the law of segregation now? So let's look at this Punnett square. So you have first generation. So your father is big T, your mom is small t. So you can see here you only get offspring is tall, but they are heterozygous. Heterozygous dominant, right? So the phenotype is dominant, the genotype is heterozygous. Now, let's see. So you cross this one again. So you cross big T, small t with big T, small t. So now both parents are big T, small t. So then the big T will separate from small t. So this is a law of segregation. They can separate. So big T separate small t. And then they also can free combine. So it means that big T combined with big T and small t combined with big T and big T combined with small t and small t combined with small t. So now, how many genotypes do you have? 3. 1, 2, and 3, correct? How many phenotypes do you have? You only have 2 because this one will show, the first one will show dominant. They are homozygous dominant. So big T, small t also show dominant. So then they are big T, small t is heterozygous dominant. So only small t will show recessive. It will show short. So therefore, you get 3 big tree and only 1 small tree. So if you look at the genotype ratio, is 1, 2, 1, right? So 1 was big T, 2 was big T, small t, and 1 was small t. Okay, so now see this one. So phenotypes, we said when they show dominant phenotypes, if you only have 1 dominant allele, so you have 2 dominant allele, sure, you are tall. If you have 1 dominant allele, you also tall. Okay, so you have 3 tall and 1 short. Okay, so the ratio of phenotypes is 3 and 1. Okay, so this is law of segregation, means that big T, small t can separate and the big T, small t can free combine. Okay. So now let's go with examples. Okay, so let's assume you already have 4 chromosomes. Your father gave you 2, A and B. Your mother also gave you 2, A and B. Okay, so let's assume that the big A is dominant and small a is recessive. So now what you can see is when we're doing meiosis, so your pair will separate, right? So your mother and your father's pair will separate. Okay, so this big A is 1 now from your dad, and small a is recessive allele from your mom. They will separate during the meiosis. So eventually you get 4 different combinations, correct? So 4 different sperm or egg. Okay, so this is here. So when you actually calculate how many different sperm you will have at least. Okay, so that's 4 if you have 2 pairs. If you have 4 chromosomes, 2 pairs, remember. Okay, so now let's look at this one. So this is a third law. They call it law of independent assortment. So we write it down in meiosis. What's the independent assortment? So in anaphase, in meiosis 1, means that the parents' chromosomes can free separate and free combine. That's why you have 4 opportunities if you only have 2 pairs, correct? Okay, so then let's assume that your mother have 2 pair of chromosomes. Your father also have 2 pair of chromosomes. So eventually your dad can making 4 type of sperm. Your mom can making 4 type of eggs. So when they do fertilization, they can recombine. So that's why you have to do Punnett square. If without this Punnett square, it's impossible, correct? So then what you get is you have the phenotypes, you have lie 3, 3, 1. So you have this different ratio, okay? Okay, so this is wrong. Why? Because the Y and R are separate. They are located on different chromosomes, so they can free separate. Okay, good. Okay, so now we face a difficult question. So I already gave you examples like you only have 2 pair of chromosomes. but most of organisms will have more than 2 pairs of chromosomes. Okay, such like you and me, we have 46 chromosomes, you have 23 pairs, correct? So now, so if you only have 4 pairs, so let's do 4 pairs. So if you have 4 pairs, you want to look at how many combinations you can have. You can do manually, especially in a test setting. So you only have short time. So of course, for only 4 pairs, you still can do manually. So now you get 16 different combinations, correct? So now what do you actually see this one? So if you have like 23, what are you going to do? You cannot use this manually. You have to do mathematically. So now let's give you a formula. So what's the formula? Formula is 2 and power m. So 2 is every given time, any gene, you will have 2 alleles because you get 1 allele from your mom, another allele from your dad. They can be the same. They could homozygous. They can be different. They could heterozygous. So here, that's 2 come from. The n is how many pairs? So we already gave you examples 2 pairs. Now I give you examples of 4 pairs. So if you have 23 pairs, then what you do is you do in this one. So 2 and the power 23. So this will give you that much different combination. Okay. So nowadays biology is mathematics plus biology. Okay, remember this one. Okay, good. So now you face a problem. What problem you have is the homozygous, they show dominant phenotypes, and heterozygous also show dominant phenotypes. For instance, this purple flower is dominant phenotypes. So you can have big P, they show purple. And you have small p, sorry, not small p, big P, purple, and small p show white. They still show purple. Okay, so from outside, you really cannot tell they are homozygous or heterozygous. So now we use this method. They call test scores to find out whether they are homozygous dominant or heterozygous dominant. So let's do this. Okay, so let's do this one. So let's say your father is purple. You don't know they are homozygous purple or heterozygous purple. So what you do is you use this white flower, the recessive, the homozygous recessive, cross with this purple flower. If the offspring is 100% purple, then this father must be homozygous. So they have got 2 alleles. They must be both big P, big P. So if the offspring, like this side, is half purple and half white, then you can show that the father is heterozygous. They are half big P and half small p. Okay, so the test course is tell you your homozygous dominant or heterozygous dominant. Okay, got it? So remember this, the purpose of doing the test course. Okay, because you from outside, you really cannot tell whether they are dominant homozygous or dominant heterozygous. You need this test course to check. Okay, so now let's finish the father genetics. So you know the father genetics is used in the plants and the trees. So you want to find out the genetic inherited, sometimes difficult. For instance, you actually doing peace plants. You really depend on the soil. You only also depend on the water, right? So the control environment will influence whether you get a big tree or small tree, or big P or small p. So that will be difficult, okay? You cannot control the weather. So now scientists actually, these scientists start using Drosophila, which is fruit fly. So they has a certain advantage to do genetics experiment. Number one, they can making offspring easily. So they can produce many offsprings, number one. Number two, the breeding season is very short. They only have 2 weeks, 2 weeks breeding season, okay? 2 weeks breeding season. Then third one is they only have 4 pair of chromosomes. So they're easy to do those course, right? So most importantly, they can easily to breed in the laboratory. So it means that you can control temperature, you can control water, you can control light. So everyone should have same condition, okay? So you will not influenced by the environment, okay? Good. Okay, now let's do mathematics. Let's introduce you Hardy-Weinberg equation. So to do Hardy-Weinberg equation, you must have a stable environment. And you have to meet these 5 criteria to do Hardy-Weinberg equation, okay? So here, so we talk about this later. We talk about this later, okay? So number one, you must have a very big population. So then this mathematics can be more significant. And also your population must be isolated from another one. So it means that if you only have 2 alleles, you always have 2 alleles. You don't have new allele coming. And you don't have mutation. So mutation can generate new alleles, right? So no mutation. And also you don't have sexual selection. And last one is, I just emphasize that you don't have environment change. You don't have nature selection by environment. So environment will not give you pressure to change. Okay, good. So this is what you need to know. Okay, so now let's understand this Hardy-Weinberg equation. So we talk about you have 1 gene. 1 gene, you must have 2 alleles. Any type of gene must have 2 alleles because on your body cells, you have diploid cells, right? So your mom gave you a SAD, your dad gave you a SAD. So you have 2 alleles, you can show phenotypes. If you only have 1 allele, you will not show phenotypes. That is clear, okay? So gene has 2 alleles. So now these 2 alleles, let's say one is P, another one is Q. So those 2 were making 1 gene. Okay, so P plus Q equal to 1 gene. Got it? Okay, so this is the law, first law. So basically on the first law, you square both sides. So then you get P squared plus 2PQ plus Q squared equal to 1. Okay? So you have 2 laws, okay? 2 laws. So we already said at the beginning, so this class we'll talk about more than genetics. So we'll talk about allele instead of a tree, okay? So now, P and Q, they're both percentage, frequency, percentage, not an absolute number. That's another thing you have to know. And the P squared, Q squared, 2PQ, also percentage, okay? But what's different between PQ and P squared, Q squared, and 2PQ? So you can imagine that. So in your germ cells, you only have 1 allele because you don't have pair anymore, correct? So you can think P and Q, the percentage of P and Q is only in your germ cells. But your body cells must have diploid. So then they have 2 alleles. Got it? So any individual body cells will have big A, or small a, or big A, small a. So all of your body cells must have two alleles. So they can be same, homozygous, P square, Q square. They can be different. 2PQ is heterozygous. Okay, so I gave you examples. So your germ cells will like P and Q, and your body cells will have P square, Q square, and 2PQ. Okay, so now let's do calculation. Start with this one. Okay, so let's see this one. Question 1. Determine the percentage of population. We talk about population, your body cells, that hybrid. So it means that they are mixture, they're 2PQ, okay? So they actually ask you to determine percentage, again, ratio. So if the frequency of allele in recessive 12 means allele recessive is 0.5. So this gives you information Q is 0.5. So according to P plus Q equal to 1, then your P will be also 0.5. Then now they ask you what is 2PQ. So your 2PQ will equal to 0.52. Okay, so mathematically are really simple, but the concepts are extremely important. So let's do another question here. Okay, so again, they ask you determine the percentage, again, percentage of the population that homozygous dominant. So the big P, if the percentage of population that homozygous recessive, what's the homozygous recessive? Q square. Q square equal to 16%. So the condition they gave you is Q square equal to 16%. What's big P? Okay, so now, so your Q square is 16%. So then the Q must be 0.4. Then P must be 0.6. Then P squared will equal to 0.36. Here, you can see they ask you determine percentage of population that is homozygous dominant. They ask you P squared, right? So now you got a P squared. Do you have a problem for this calculation? No. Okay, good. So let's emphasize. Okay, so how you use on this in your body? Just think about it. All your germ cells only contain one allele. They can contain in big B or small p. Do they show phenotypes? No, they just one single cell, correct? And think about that your body cells must have two alleles because they have so many genes. They will show phenotypes. So your body cells will show phenotypes. Okay, so you have dark skin, you have green eyes, or you have black eyes, correct? That's the phenotypes. The phenotypes only show on your body, not your germ cells. Okay, now let's do this question. Your turn, please. Good. So now let's go through this together, right? So the 9 is A, B is D, and 19 is D. So 19, you actually have to remember here is talk about percentage. If they give you not in percentage, you have to convert to the percentage. For this one, they give you 1,000 people, and 90 have a blue eyes, means that the not percentage. So you have to calculate the percentage. So then the percentage for Q square, because they show phenotypes. So there must be Q square, P square, 2PQ, right? So then you get this percentage is 9%. So Q equal to 0.3, and P is equal to 0.7. So now they ask you hybrid, heterozygous, brown. So 2PQ will equal to 42%. Okay. So now let's give you another question here. Calculation again. So one thing, before you do this question, we have to go through the RH blood. Everyone knows that you have ABO blood, right? So that's when you have a birth, they're showing on your birth certificate. But there's another blood type, which is RH positive or RH negative, okay? So RH positive is dominant. RH negative is recessive, okay? So now you can do that. So you can type on the screen or you can answer in the chat as well, okay? Okay, so let's go through together. Okay, you are right. So you follow this Hardy-Weinberg equation, right? So they actually told you that, see this one, 6%, 16% of population is Rh active. So then you actually have 2,000 students. So what you can see here? So you have Q squared equal to 16%. And so Q will be 0.4 and P will be 0.6. So you have 2,000, the whole population 2,000. So let's work on here. So 2PQ equal to 0.48, 48%. P squared equal to 36%. So the Q squared, I already told you, is 16%. So you multiply this percentage to the whole population. So you get big A, 720, big A, small a, 960, and small a is 320. Okay, so simple calculation. Okay. So now let's introduce pedigree tree. So this pedigree tree is used to retrieve your family genetic disease. So they can determine whether you are dominant or you are recessive or you are sexual linked disease. Okay. So here, see this one? So what is shown here? The round figure is represent female. The square figure represent the male. Okay, the solid one is diseased. The empty one is no symptoms. Okay, so other one, your body cells, you should have this pair. So you have 3 pairs, sorry, 23 pairs. You have 23 pairs, right? So therefore, here, whether they show dominant or recessive, is really depend on, number one, the combination of the genotypes. Number two, whether they're dominant or recessive, or whether they are on the sexual chromosomes or they are in the autosomic chromosomes. So only the 23 pairs, the 23 pairs is X and Y, and all the others is autosomic. Okay. So here, see this one? Okay, so what you can see here, this is autosomic, means that the disease genes is on number 1, number 2, up to number 22. So this is called autosomic. And another one is sexual linked. The most common one is X linked, means they are on X. Okay, they could have a Y linked, means they are located on Y, but extremely rare. Okay. So now let's talk about most common X-linked disease. So for the X-linked disease, most common one are recessive. Okay, you probably see this recessive X-linked disease quite often in your life. For instance, okay, so this one, so you walk on the street, you can see a young man, probably only 30 years old, they don't have any hair. So this is a kind of genetic disease we call baldness. Okay, so this is the disease. So you also can see this person rarely. Okay, so they actually have muscular dystrophy. Okay, so when they very young, So before age 22, the muscle lose the ability. So they have to sit on the wheelchair. The parents have to take care of them. So that's another one. So another one are very common in African countries. They call it hemophilia. So if they cut their fingers, normally you don't worry, right? You cut the fingers and it will stop. The bleeding will stop. The bleeding will stop automatically. But for this patient, they cannot. They just continue to bleed. So you have to send them to the hospital. Okay? So this is called hemophilia. Another one is colorblind. So some of children, they cannot distinguish the orange, red, and green. So especially red and green, they cannot see. They cannot see difference. Okay, good. So look at this one. Okay, so look at this one. So let's see, this is the father. The father is X and Y. So we said the disease genes is on X. They are recessive. Okay, recessive sex-linked disease. So you can see father is normal because they got hair, right? Mother is normal. The mother appear normal, but the mother can carry a disease gene because the disease is recessive. Okay, so the disease is recessive. The mother is carrier. They are not show any symptoms. So of course we follow these 3 laws. So they will separate, they recombine. So then you can see the children. So the orderly daughter show normal, no symptoms. But one of the daughter is carrier. One is 100% normal. How about the son? So you can see this son is absolutely normal. They got hair. So this one is no hair. So you get 50% of some show the disease. Okay. So why this one show the disease? Because X and Y are different chromosomes, completely different. They carry different genes. So the X cannot cover Y, Y cannot cover X. So even you only have one disease gene on X, they will show up because no one will dominate them. Okay. Okay, so from this experiment, you can see that the X-linked recessive disorders predominantly affect men. Okay, so they will affect the man predominantly. The woman will not affect too much. Okay, so same thing for the hemophilia. Okay, good. So now let's do this challenging question, please. So one thing you have to remember that any child only have one dad, one mom, biological mom, biological dad. You cannot mix those two, okay? So any given child, you only have one condition, one biological mom, one biological dad, okay? So look at this question. You have to do Punnett square. I already mentioned to you, I already remind you, your brain is not good enough. Okay. So you have to do Punnett square. Have you done Punnett square? Do Punnett square, okay? Okay, so let's go through together. So answer is E. I noticed someone answer C. Okay, so now let's see this one. So you have father. So father is disease, father. So this is X-linked disease. So let's assume that X-linked recessive disease. So X on the father is abnormal. Okay, for sure. So which one is like that? Like this first one? So they said the mother is non-symptoms, no symptoms. Mother is normal. So here, mother is absolutely normal, no carrier. So therefore, you get children on here. So none of the children get disease. Okay, so you can see all of the son normal because they inherited the mother's X. Mother X is absolutely normal. Okay, so another one is the daughter. So the daughter not show any symptoms, but all of the daughter are carrier, correct? So this is one situation. Another situation is the father is diseased. The father X is abnormal, mother is carrier. So one X is normal and one X is diseased in the mother. So when they cross, you find that, yeah? So you get daughter, one is carrier, one is diseased. And son, one is normal, appear normal, and absolutely normal in genotypes, and one is diseased. So which one is match? E, correct? So if you answer C, it's wrong because you said approximately 50% of the daughter will have X-linked disease. So then there must be this, the second condition, correct? Okay, so if you choose the mother is carrier, then the son, the half of the son will have disease. So that's why this one is not right because they're not match C. And also I told you that you cannot mix those two conditions because every individual are unique because you only have one and one biological father and mother. Okay, good. So now let's continue. Okay, so let's talk about Y-linked trials. Y-linked trials are extremely rare. Okay, so also they are absolutely dominant because we don't have anyone cover Y. Y and X are carry completely different genes. Okay, so then if father got disease, all of female in the family is normal, no problem. But all of male got disease, okay? But extremely rare, okay? Good. So now let's talk about something very interesting, okay? So what is interesting is here. So in the animal, especially in the mammals, so you have here, you have the female, you have 2X, correct? And male, you have only 1X. So in the female, so this is only happening in the female. So if you have a 2X, then Y is not working. They take off. They take this off if you have 2X. So this is happened in the mammals. So you can see here. So this is the cat. So the fur color is on your X chromosomes. So they can be black. See this one? They also can be orange. Okay, so there's two different alleles on the X. So let's see. So let's format. If you both X get black, of course you black format. If both orange, okay, you are orange format. But if one is black and one is orange, then it's tricky. Why? Because Y is not on duty, right? So then another one is dominant. So let's see. So this is random. So let's say this region and this region, okay? So this region, you can see orange. So here it's like this one. See this one? So this one, you can see is orange X on duty. The black one is taking break. So that's why they show orange. But this black region is the black on duty and the orange take break. So then they will show black. So this is random, okay? Random for individual cells. So you can see this region is black because the black acts on duty. So this area is orange because your orange on duty, okay? So this only happened in the female. So how about the male? Male only, you have only one chromosome, one X chromosome, one X chromosome, one Y chromosome. So they have to on duty, okay? They cannot take in break. So therefore, if the orange, if the male, if the boy has orange X, they show orange. If they have black on X, they show black. You only have simple color for male because you only have one X, okay? Good. Now, let's do this question, please. Don't forget your Punnett square, okay? So do this question, please. Good. Answer is D, because we said the boy cannot be codical cat, right? They cannot be multiple color. Good. So now let's talk about nowadays genetics. So the genetics more is actually has more complicated than the further genetic theory. So now let's talk about number one, incomplete dominant. So look at this one again. So you have white flower, yeah, you have red flower here. You mix these two, so you get pink flower, okay? Pink flower, got it? So then if you cross these two pink flowers together, so the egg is pink, the sperm is pink, then you get those three color. Okay, so you get red, pink, and see this one? No white. So you get new color come out. So that's incomplete dominant. Means that no one is dominant for others. Okay, so then you get a mixture. So another one is called co-dominant. So this is another example. So you get white cow, mixed with brown cow. So you have upstream as both colors. So white and brown. You do not have a new color. No new color appear. So that's the key difference between incomplete or co-dominant. Incomplete, they has a new color. Pink is a new color. And the co-dominant means they are dominant. They cannot mix another one. So the offspring will have two colors. Okay, got it? Good. So now in the early video, we said a lot of phenotypes are called polygenetic trial. Means that they controlled by not only one gene, multiple genes, okay, a lot. But most of commonly used as examples in your textbook is your skin color, eye color, and also your height. So let's think about this way. So such like you have 4 siblings in your family. No one is exactly high. And no one has exactly same skin color. So if you look at the eyes, no one has the same color in the eye, okay? So that's because each of these genes contribute part of the color, okay? So this is called polygenetic trial, okay? So you also have multiple alleles, but the multiple alleles is only controlled by one gene. So each gene we said is fixed. You only have 2 alleles. So now the multiple gene in our body, such like ABO blood. Okay, so how many alleles you have? You have A, you have B, you also have O. So O stands for I. So we use I for the O. So A dominant, B dominant, and I recessive. So any given time, you only have 2. So you can choose A, so you got A blood. If you choose A and I, you also have A blood. If you choose A and B, if your parents give you A and B, then you are AB blood, okay? So if your parents give you B and B, you are B blood. If your parents give you I and I, you are O blood. But at any given time, you only have 2 because only have 2 chromosomes give you alleles. Okay, so then you have A, B, and I. Okay, so you have 4 different blood types: A, AB, B, and O blood. Correct? So now let's do this question. Again, do not forget your Punnett square. Actually, I did the Punnett square for you, but you should be able to do your own Punnett square, please. Okay, so now let's look at this question. So your answer is D. So let's go through together, okay? So let's say your mother is fixed, A and B, A, B, right? So your father is flexible, and the children, one is A, another one is A, B. So let's see. So let's assume that the father is A. So I give AI, the heterozygous, because AI including AA, correct? So if you do AA, then you only do this half, correct? So then you can see if you do AI, they were including AA. So then the children can be A, can be homozygous A, heterozygous A, AB, and the B. Good, no problem. So if father is a B, so let's give you BI, same reason. The BI including BB, because BB only this half, okay? So then if father is B, then you can have AB, AI, BB, and BI. So you can have A and AB, correct? No problem. So how about the father is AB, and the mother also AB. So now you can see the children can be A, AB, and BB. That's okay too, correct? So let's say the father, is that possible like O? So O is II. So then the children can have A or B, not AB. So your choice D is right. Okay, good. Okay. So now I'm going to give you a 10 minutes break. Then after break, then we continue to talk about linked genes. Okay, so 10 minutes break. So let's start again. So let's talk about linked genes. So what's the linked gene? So you know that your chromosomes are very long. So they contain many genes. They contain thousands of genes. So all of these genes on the same chromosomes, they call linked gene. Okay? So all of these on the same chromosome is called linked gene. Got it? Okay. So for instance, the gene 1 and 2 are linked genes. So in here, so this is not linked because here, so gene 2 is on the big one and gene 1 is on the small one. And also AB is linked, okay? So remember, any genes on the same chromosome, they are linked genes, okay? So this is a copy from the textbook. Sometimes confuse student. You only need to remember is if at that moment, those two genes on the same chromosomes, they call linked gene, okay? So if they linked, so they should move in together in mitosis or meiosis, correct? So then, see this one, you should treat them as a single gene. So for instance, you cross A, B, B with small small b, because they are linked. So you should just treat as they only have AB. AB is one gene. Okay, so you can see the phenotype is big A, big B, small b, because they only have one single type. So you can see if they cross with A, B, B, see this one? So then they got two, right? So you really need to treat as they are single gene. Okay. So another concept we talk about is that here, see this is two concepts, okay? So number one is you talk about they have one disease, but many symptoms, such like sickle cell disease, right? We already talk about sickle cell disease because you have this sickle cell, so they breaking down. And also they lose the hemoglobin ability to touch to oxygen. So they actually have multiple symptoms. So they block your blood vessel too. So you have all these symptoms, okay? But you only have one disease. So this is the concept. Another one, so this is another concept. So they call epistatus. So these are much more complicated. Means that the gene is not controlled by themselves, it's controlled by another gene. Okay, so now let's talk about this one. So we talk about some disease base, okay? So what of we did before for the cell division is normal. So now let's see some abnormal. So this is called non-disjunction. So here, what you can see is during the meiosis, you can see in the meiosis 1, you should separate the pair, correct? So this, the right one is correct one because they will separate, okay? They'll separate to the two different cells, but this side is wrong. See this one? Because this part hasn't separate. This one did not separate. This part did not separate. So this is non-destruction happened in meiosis 1 for this one. So then this one has 3 chromosomes. This one only have 1. So then end of result, that you have 2 cells with 3 chromosomes, 2 cells only 1 chromosomes. Okay, this one. So you can see they have problem with meiosis 2 because meiosis 1 is normal. Each one only have 2 chromosomes. In the meiosis 2, this one is normal. Okay, this one's abnormal. So one got 3, another one got 1, and these 2 are normal. So this is called long disjunction in meiosis. During the mitosis, you also can have this problem, but mitosis, they only have a severe consequences right from beginning. When you are in the zygote stage, you are one cell. So then if you have this long disjunction happened, you have consequence. You have half cells has more than 1, another one has a less 1, right? Okay. So for the long disjunction, especially the meiosis, you can end up with polyploidy, means that you are not diploid. You have 3 set or 4 set. Okay, so that's the consequence. Okay, so now that's more complicated situation in the genetics. They call imprinting. Means that although your gene showing normal, you have dominant allele, but it didn't show dominant. Okay, so that's because your internal environment. Okay, so now we just learn for the cell structure, right? We learn cell structure. We said, of course, your nucleus has all of the chromosomes, but don't forget you have chromosomes or DNA in mitochondria, in chloroplast. So let's use mitochondria, okay? So mitochondria, you have circular DNA, remember? Just exactly like bacterial DNA. And so this can be abnormal. So then in the human, if you have this problem, then you could end up disease. So this is eye disease. Okay. Another thing is very interesting. So when you doing fertilization, then the mother gave half genetics material and also gave all of cytosomes. So your father only gave half of the DNA. So what's the consequence? All of mitochondria is from your mom. None of mitochondria from your dad, because your dad only gave you half of nuclear DNA. Your mom gave you half nuclear DNA as well as mitochondria. So then what mitochondria from your mom? Okay, so now you can see here. So let's see. Okay, so this is one. So let's see this left side. So your father is unaffected. Your mother is got the mitochondrial disease, mitochondrial genetic disease. So you can see all of the children got disease because all of the children's cells, the mitochondria is from your mom. So let's look at this one. Okay, so you have here affected father, you have a normal mother. So you can see all of the children are normal because your mother gave all of the mitochondria to the 3 children. Okay, got it? Okay, so that's the special pattern for mitochondria inherited. Okay, so remember we talked about crossover in the meiosis. Okay, so now let's calculate the crossover frequency. So individual crossover is not fixed. It's not guaranteed happen. They happen randomly. Okay, so that's the problem. So sometimes problem. Okay, so that's also the consequence of why everyone is different. Okay, this is one of the reasons you have genetic variation. We did this one last time, correct? So now let's see. Okay, So let's see, this is just one chromosome. Okay, so you have two, right? So you have haploid chromosomes. Your mom gave you this one. Your dad gave you this pair as well, and they're making a pair. So let's see, you have crossover. Crossover is between your mother's chromates and your father's chromates, correct? So now, when you're making secondary cells, when you do meiosis, they have crossover. So let's see. So we actually see this one. So C and D, you have crossover about 10%. And between B and C, you have another 10% crossover. And between A and B, you have 10%. Okay, so then if you look at C and D, you have 10%. Okay, C and D only 10%. But the B and the D, you have 20% because you cross over once and second time. Okay, so that's B and D. So that's B and D, you get 10% plus 10% equal to 20%. So let's look at A and D. So A and D, you get 10, 10, and 10. So then AD, you get 30%. So what's the conclusion you can draw from this picture? So if you two genes have short distance, then the crossover rate is lower. So if two gene has a high distance, then the crossover is higher. Okay, got it? So if you are more distance, you have high crossover, you can cross off once and twice in a different location. If you are very close, you only have a lower chance. Okay. So let's do this question, okay? So we already talk about concept. So if you are more distance, you have high crossover. If you have very close, if two genes are very close, then you actually have a low crossover rate, okay? So we talk about A, B, C, D. Now I change the word, E, G, F, H. So everyone start with E. So no doubt start with E. So everyone compare with E. What do you think? So the number is crossover rate. Again, if you got the highest one is 22, means that they have long distance, correct? Good. So let's do this one, please. Okay, so now let's go through this question together, right? So let's say we got 22. 22 is the highest number, means high crossover. So high crossover means a longer distance. So E and F must have a longer distance. So you put the F in the end. So who has a short distance? E to 12, right? So the 12 is a short distance. means that E and H are very close. So you put E, H together. Then something between is G with 17. So then G is shorter than F, but longer than H. So you put E, H, G, F. Okay, good. So let's do calculation now. Okay, so I cover this one. And in a real AP or competition, you have only 1 minute or at the most 1.5 minutes to finish this question, okay? Think about it. So one thing I have to explain when you start is here. So if they talk about crossover, means that originally they linked, okay? So if they talk about crossover, means they must link before, then you have crossover. If you are not linked, there's no way to crossover, right? Okay, although at this question at the beginning, they didn't mention that you are linked between G and N, but because they mentioned the crossover, then GM must be linked. Okay, got it? Now, do it now. Okay, so let's go through together. Okay, so let's assume so this side. Okay, so let's start with this one. So two genes are linked. So which two genes are linked? Is a big G linked to big N, means that those two alleles are on the same chromosome. So big N on the same chromosome as big G. So small g is same chromosome as small m. So this is linked. Okay. So if they're 100% linked, then you should treat them as one gene. So then when you do this Punnett squares, you put G and M together and small g and small n together. So you cross with G and N. They ask you to cross with GG and N, but the lowercase one. So then what you get? So you will get half of big G, small g, big N, small m, half. And half small g, small n, small n. Right? Got it? So let's see how many phenotypes. You have 4 phenotypes this time. Not 2. Not 2. So then you must have here, you still have a big G linked to big M and small g linked to small m, but you have crossover. So the crossover is happened. So now they ask you to calculate what's the crossover rate. Easy, okay? So which one you think is without crossover? So A and B are without crossover, correct? So the big A, big B, so these 2 categories are not crossover result. C and D are crossover result. Okay? So when you actually calculate the percentage of crossover, so you add all this up, everything, then this is the total number. So what's the crossover number? Is this 2, correct? C, 190 plus 180. So what you get is here. So you add these 2 to get 374 divided by 2,284, then equal to 16.4%. Okay, the calculation again is very simple. What is most important, you have to know the concept. So see, I gave you an illustration. So you have 2 chromosomes, correct? So 2 chromosomes, this is a pair. So let's say the blue one is your mom. So your mom will give you a big G, big N. Your dad is here, mixture. So your dad gives you a small g, small n. Okay, so if you don't have crossover, then they separate. So if you do have crossover, okay, so you got a mixture. So then you get your mom is big G linked to small m, and your dad is small g linked to big N. So that's a new one. But majority is not being crossed over. So then you have this one as a major phenotypes. This one, you have a lower percentage. Okay, got it? Good. Okay, so you have more questions, more difficult questions to think about it, to review this concept, to do at home, so you can check your result with my result in your lecture notes. Okay, good. So now, so that's another question. Okay, so think about it. Okay. So now let's talk about the genetic disease. So we already talked about a few diseases already, correct? We talk about sickle cell disease. Okay, so this cystic fibrosis is also kind of common disease. It's also recessive. Okay, So Huntington disease are dominant disease. So they all autosomic disease. Means that the disease allele is in number 1 or number 2 up to 22 chromosomes. So we already discussed sexual linked chromosomes disease. So most common one we talk about is X-linked recessive, right? So hemophilia, breathing problem, colorblind, you cannot distinguish red and green, and dystrophy, right? And also the light disease like borderless. Okay, so now the orders is the number, the chromosome number is normal. Okay, so now I'm going to talk about if you have different chromosome number. Okay, what the disease are. So let's start with XXY man. So this is man, but they get 2X or even 3X. The cause is non-disjunction, okay? So look at this one. So you can see here is the XXY man, okay? So look at this one. So let's look at meiosis 1 non-disjunction. So you get X and Y in your body cells, right? So your cellular cells will start with XY. So they're doing meiosis 1. So in this time, you can see X and Y, and they move to one cell. They didn't separate. So this one has no sexual chromosomes. So then they divide again, meiosis 2. So you can see this 2 sperm has XY. This one has no X, no Y. So this one will have how many chromosomes? 24, correct? Because they have X and Y. So this one only have 22. So now let's suppose this XY sperm mix with XX. So they generate XXY man. So this is XXY man. So this is the chromosomes picture. So they only have this one abnormal. Other things are normal. So you have 2X and 1Y. So what symptoms for this man? So this man is man. So they have texts. And they show as a man, a period, man figures. But they have a breast developed. And also the hair is very little. So they don't have beard, maybe. But the body hair is really, really soft and few, okay? So this is the egg, okay? So the mother, okay? So you can see here. So this is 1. So when they do meiosis 1, so you get 2 eggs go to 1 germ cell. Then they divide again. So the 1 egg has double X. Okay, so if you get this egg, then you cross with normal Y sperm, you get the XXY man. Okay, so you also have this one, see this one? So in meiosis 2, your chrome T hasn't separate. Then again, you get 2X. So you still have XXY man. So you get 47 chromosomes. Okay. So sometimes XXXY Okay. So another disease called Turner syndrome. So Turner syndrome is this little woman. So she's not 5 years old. She probably 20 years old, but she appear like 5, 6 years old. Okay. So what happened is during the father's meiosis, again, so you can see in this one, that they did not get any sexual chromosomes. So then this sperm do not have any sexual chromosomes. So when they cross with a normal X egg, they actually become the Turner syndrome. So this is a woman, but very short. They also can happen in mother's non-disjunction meiosis. Okay, same thing. If this egg without any eggs, then when they got the egg sperm, they got Turner syndrome. Okay, so again, they only have 45 chromosomes. So another common disease is this Down syndrome. So I'm pretty sure everyone sees these children on the street or somewhere. So because they have quite a lot, so we have Down syndrome association because this is really a problem. Okay. So this child has a multi-symptoms. So the brain is not fully developed. Okay, so they have eye problem, you have hearing problem, you have a genetic cause the immune deficiency, all these different department, the problem. Okay, the problem with this one is again, they have abnormal number of chromosomes. They have 321 chromosomes. Okay, 321 chromosomes. So this is consequence from the non-disjunction. They can happen in meiosis, or they can happen in the beginning of mitosis. Okay, meiosis, mitosis. Of course, meiosis probably is a major cause. Okay, so now let's do some question. Okay, so question time, please. Okay, your result. So 1C, and 2C, 3D, the same one? No. Okay, so now let's look at this one. Okay, so, okay, this is, oh, same. Okay, good. So I didn't give you 3, right? I didn't give you 3, 1. Oh, how funny. Okay, so let's look at this one. So 1C, 2C. So if you haven't done 3, think about 3. So 4 is D. Okay, 4 is D. 4 is D. So let's see whether you did D or not. Why you got a C, dear? So 4 is D and 5 is A. Okay, so why you got this one? Pink. So let's do together, okay? So you cross with 2, right? How can I get this pink? So here. That must be wrong because when we're doing this one, so let's do this together. Why I put this wrong? Okay, it's good. It's good I get it wrong and you correct me. Yeah, good. Okay, so let's import a table. Okay, so let's do together. Okay, don't worry. You got 2 pink, right? So let's say 2 pink. So you get 2 pink, right? R, W, R, W. So R, W. Okay, so they are incomplete dominant, I guess. So you get RR will be red. Okay, red. You get RW, which is pink. You get RW, pink. And you get WW, white. Okay, so now, so let's look at this one. So, okay, it's a 50%, right? So 4 is C, good, good. Okay, so 4 is C, so 50%. So pink is 50%. So 5 is A, right? You don't have problem because you did Punnett square, correct? Good. Okay, so now let's do more question. So we're done this one. So now let's do those questions. So if we have problem C. Okay, so let's have a look. So 6 is E, 7 is E, and 8 is A. So you all get it right. So these side are difficult. Why? Because they are symptom. Okay, so let's go through together then. So 9 is E, 9 is E, and 10 is B, 11 is A, and 12 is C, and 13 is D, and 14 is C. Okay, so let's do some of those questions. Okay, so let's do those. Okay, so now let's have a look. Okay, so number one, you have to decide whether they are dominant or recessive. So if it's dominant, then every generation should have disease. So for this one, the both parents do not have disease. So therefore, this must be dominant. Okay, so if your decide is not dominant, then you will cross B and C because they talk about dominant, right? So the B and the C is a dominant, but this is not dominant. So second, you have to decide whether they are autosomic or sex-linked. So let's say you assume it's sex-linked. Okay, so you say it's X sex-linked. So let's see. Okay, so if it's X sex-linked recessive disease, means that the disease chromosomes is the X. So the alleles are X. So therefore, the mother must be carrier. Correct? The mother must be carrier. And father must be normal because if the father is carrying disease alleles, they must show symptoms. So therefore, the son can get disease because mother is carrier, no problem. So daughter, see this one? The daughter get disease. So this is impossible if they are X-linked recessive disease because they have to have a both allele carry disease gene. So then the mother can give one, no problem. The father also have to give one. That's impossible because we already assume that this is X-linked recessive disease. So then, for this case, the daughter cannot have a disease because there is the two allele carry recessive allele. Okay, so now you cross this one, right? You cause a sex-linked recessive disease, right? You cause B, C, and D. So E is impossible because this one only show the normal inherited, not incomplete inherited. So the answer is A. So let's say you already decide this is autosomic recessive disease, then see the C, which one is this one? You see, so in order for any of the children, regardless is a daughter or son, they must have two chromosomes that disease allele. So the father can give one, mother also have to give one. So mother must be carrier because they didn't show disease. Okay, so answer B is correct. Okay, good. So now we are going to move new chapters. We're going, so you have more questions, you can do practice at home. And we're going to move to molecular biology. We're going to talk about molecular biology, but before I talk about molecular biology, I give you a 5 minutes break because we have extra 15 minutes lecture. Okay, so I give you 5 more minutes break, then we can continue. Okay, so let's start with a new chapter, molecular biology. Okay, so let's start with the biggest experiment in this 50 years. So the biotech, Human Genome Project. So we started this project in 1987. Then in the 2002 or 2003, we got first version of our human genome. So we sequence our whole genome. Takes so long. So what result we get from this experiment? Number one, we found that although you and me will never meet, but our genome has a 99.9% same. So only 0.1 difference amount of all the people. Number two, in our genome, only 2% of DNA containing gene, which is an instruction to making protein. So you may have a question, say, how about another 98 or 97% of DNA do? Scientists have the same question as you got. So in that time, scientists really panicked. They said, look, we have 97% of junk in our DNA. Is that true? It's not true, okay? So although those 97 or 98% of DNA is not coding sequence for protein, but they play very important role to regulate the transcription, okay? So now let's start with the first one. So we talk about the DNA structure, okay? So someone may say, oh, I already know DNA structure, but be careful though. And we're going to do competition or AP. So you have to go to more deep understanding for DNA structure. Okay, so now let's again give you this very short video to give you, to remind you, okay? You may already learn that, but we have to know this one again. So now let's look at this one.
All cells require some form of instructions to be able to function properly. They need guidelines, rules, codes for making materials in the cell, and that code is DNA. DNA stands for deoxyribonucleic acid. D for the name of the sugar, N and A for nucleic acid. DNA contains the information that determines inherited characteristics. It has the code for making proteins. DNA is found in the nucleus of eukaryotic cells and in the cytosol of prokaryotes. If we take a closer look at the chromatin inside the nucleus, we can see the structure of the DNA. The DNA has repeating subunits, and those subunits are called monomers, or nucleotides specifically. The nucleotide has three main parts: a phosphate group, a sugar, and a nitrogen base. In DNA, the name of the sugar is deoxyribose, which is part of DNA's name. And there are 4 nitrogen bases in DNA. The bases are thymine, adenine, cytosine, and guanine. 2 of the bases are purines, which have a 2-ring structure, and 2 bases are pyrimidines, which have a 1-ring structure. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. I remember the pyrimidines are the bases with a Y in their name, just like pyrimidine has a Y in its name. A purine always pairs with a pyrimidine, and the slanted shape of the DNA molecule causes it to form a spiral or helix. Because DNA is double-stranded, we use the phrase double helix to describe its structure. There are 4 scientists who are credited with discovering the shape of DNA, and they are Watson, Crick, Wilkins, and Franklin. All of them received a Nobel Prize for their work, except for Rosalind Franklin. She died before the prize was given. Each of these scientists played a role in piecing together the structure of DNA. They learned that along the sides of the molecule was a backbone made of alternating sugar and phosphate molecules. On the inside, like the rungs of a ladder, are the nitrogen bases. Adenine and thymine form hydrogen bonds together. Cytosine and guanine form hydrogen bonds together. To help me remember which bases link together, I think of writing the letters. A and T both use straight lines. C and G use curved lines. I also know that A and T have 2 hydrogen bonds, but C and G have 3 hydrogen bonds by saying AT2, CG3. Silly things like this are actually a great memory tool. Strands of DNA are said to be complementary to one another because A will always be with T and C will always be with G, based on the number of hydrogen bonds that they want to make. You can predict the complementary strand if you know the other strand. So let's use all of that knowledge about DNA and see if we can identify the parts of this blank DNA molecule. The easiest to label are the deoxyribose sugars and phosphates. They make up the outside of the molecule. Deoxyribose is a pentagon shape, and the phosphate is just a small molecule in between. Next, we need to remember a rhyme, AT2CG3. So the nucleotides with 2 hydrogen bonds must be A and T, and the ones with 3 are C and G. To tell which one is which, you need to know that pyrimidines have 1 ring and purines have 2. So cytosine and thymine are the pyrimidines, so they're the 1-ring bases. Adenine and guanine are the 2-ringed bases. And now you have a labeled DNA molecule. Thanks for watching this episode of Teacher's
Okay. So now let's back to our lecture notes. Okay. So let's review. So your nucleic acid are polymers. So they including DNA and RNA. So your DNA has a double strand helix. RNA has an irregular shape, whereas mRNA has a linear shape, and RNA and tRNA are very irregular. Okay. So look at this one. So remember we talked about the protein also has a helix. So the second structure is called alpha helix. So the second structure also including plant sheets. Okay. So remember also the here the DNA including nitrogenous base. So A, T, G, C. So in the middle. So they pair each other, right? Okay. So A pair with T with 2 hydrogen bond and G pair with C with 3 hydrogen bond. So this one, cake, now sugar, the 5 carbon sugar, 5 carbon sugar, 5 carbon sugar, right? So what you can see is here is the link with a phosphate group. So what is a 5 prime? So the 5 prime means that your phosphate linked to 5 carbon, number 5 carbon on the sugar. What do you mean by 3 prime? So it means that here you have number 3 carbon. They can potentially link to a phosphate group. So we said you have 2 strands. So each strand is linked by phosphate. So the phosphate link individual nucleotides together. We also talk about antiparallel. So antiparallel is one direction is 5 to 3. The opposite direction is 3 to 5, correct? Okay, so now let's look at this question. Okay, so now the answer is B and C, right? So C is absolutely right. We just talked about for the DNA, it's double helix. For protein, you have alpha helix. Another thing we talk about is elements to making DNA is what? Is nitrogen space. So you have N, correct? So no kind of one. So you have M, you have plenty of N, you have O, you have C, you have H. You also have phosphate, right? So that's all nucleotide. How about protein? So in the normal protein, you have N terminals, you have M. T terminals, you have C. You also have R group, right? So you don't have phosphate group, okay? So that's why this answer is B. They both contain nitrogen, no problem, okay? Okay, so now let's back to review this nucleotide very quickly. So we have RNA nucleotide. See, this is RNA nucleotide. So the reason they called ribose because on the second carbon you have OH. This is a DNA nucleotide. So on the second carbon you don't have O. So that's why they call deox. So that's the sugar. Okay. So this DNA RNA, so you have this called nitrogen space. Okay, so nitrogen space, you have CAGT for DNA, RNA, you have CAG, but also U. So U and the T are special for the RNA and DNA respectively. And then you can divide those nitrogen space in two types. So number one is called a pyrimidine. Pyrimidine only have one ring, okay? So they are C, T, and U. Purine has two rings, so they are A and G, okay? So we just talk about this nucleotide, okay? So nucleotide is very important structure. So this nucleotide can make DNA, RNA, and they also can making energy molecules, ATP. ATP is you have RNA nucleotide and also have a 3-phosphate. They're also making the message, so CMP. Okay, so these are extremely structures, so I highly recommended to remember this structure. So look at this one. So we write it down this one, just quick review. So the DNA RNA is a nucleic acid. You have a nucleotide as a monomers. So sometimes some textbooks say nucleoside. So nucleoside, you have base, you have sugar, but you don't have phosphate. Okay, so that's your DNA. How you actually making this here, the bond, covalent bond. So this is here. So you can see, so this is 5' end, right? So your nucleotide is here. So let's making this group. So this is here. So this is one nucleotide. So this is another nucleotide. Okay. So you can see here, see this is another nucleotide. So you can see here, see this one? So you have this phosphate group. So the phosphate group, primarily they link to 5' end of carbon. So this is 5'. So when they link to another nucleotide, they link to 3'. So the phosphate is a link to your individual nucleotide together. Okay, so this is the one. So that's why this is called a 5' end, because you got a phosphate just linked to 5. So this one is 3' end, means 3' end and 3 carbon and linked to another phosphate. Okay. So look at this one. So this is just opposite. So you can see here. So this is, let's change color. So you can see this is one nucleotide. So this is another one. So this is a nice one. Okay. So this side is 5' end because your phosphate links to 5 carbon. So when you actually do DNA replication or DNA synthesis, you actually add in this phosphate to the 3 carbon. Okay, so this is how you do it. So from this one to this one. Okay, good. Okay, so now let's talk about your nitrogen space. Okay, so we already talk about you have 2 types. One ring is TCU, okay? So, or you can see CTU. So this is pyrimidine. Purine, you have AG, okay? AG, you have 2 rings, okay? So in the DNA, they have pair. So your A pair with T, G pair with C. So in the DNA, you have half purine and half pyrimidine. But in RNA, you cannot say because they are single strand. Okay. Remember this one? So they pair each other, right? So now let's do this one. So you can do this one biologically or calculation. Okay, so let's do calculation first. So answer is C. So they already told you that here, so G is 32. So remember that's DNA. So if you have G is 32, then C must be 32 because you have two pairs. So then they will cost you 64. So remaining must be A and T, right? So you have 18 of each. So that's a simple calculation. Or logically, without calculate. So A pair with T, A, T pair. So then, come on, equal amount. And G, C pair, equal amount. Okay? So then you have half G plus T, half C and T, and half G and A, correct? So half C and T and half G and A. So 50%. So two ways you can do that. Okay, so now you also notice that A pair with T is 200 watt. G pair with C, 300 watt. So who is stronger? G pair with C. GC bond are stronger because you have 300 watt. Okay, so let's see RNA. So RNA has a three ways differ from DNA. Number one, the sugar is rainbow. Number two, they are single strand. Number three, they have uracil. Okay, so now let's pair again. So when they actually copy information from your DNA, they have to pair. So still G pair with C, no change. But A pair with U, A pair with U, okay? A pair with U. So here, see A pair with U, A and U pair. Okay, so now let's do this question now. So this is too easy, okay? So now, I think you can do this by yourself. So now, let's talk about the melting point. So melting point, let's talk about definition first. The melting point is defined as temperature at which half of DNA strand are separate or random. So half separate. So here, this is really depend on the length of DNA molecules or whether you reach in GC pairs, because we already said the GC pairs are stronger because they have 3 hydrogen bonds. So let's assume that you have same length. The DNA length is the same, but your GC contents are different. So let's say here, so you have a lot of GC bonds. So you have a lot of this GC. So if you want to separate half of them, you need a high temperature. You need 110 degrees. If you actually have a more AT, then AT is 200 bound. So they are easy to break. So therefore, you can use 70 degrees to separate half of them. Okay, so remember this concept? GC are more stronger bonded because they have 3 hydrogen bonds. So you have to have more energy to break it up. Okay, so now let's check your concept. Yes, you are right. Okay, I say C because they have all CG, CG, CG. So they will form GC pair. So they have more hydrogen bond. So you need increased temperature to get this separate. Okay, good. Okay, so let's review again. So for eukaryotic cells, you have a linearized structure. For your chromosome, so your DNA is associated with histone protein. Your DNA is linearized structure. So your genes may contain introns, non-coding sequence. Remember, we just talked about, you only have in human now, you have only 2% of DNA are coding sequence. So 98 or 97% are introns. So your DNA is in the nucleus. Whereas precancer cells, we talk about bacteria. So you have DNA is not bound to histone protein, so they are free. DNA is circular, okay? And then they don't have a multiple chromosomes, you only have one chromosome per cell. And the gene do not contain intron. And also they don't have duplets, okay? So we end up today. So, okay, you have a good weekend. Bye-bye.